Our second speaker, whose name I will, again, try to pronounce correctly, Dave duVerle, is worthy of applause. Yes. Dave, as he is commonly known, which is because it's his first name, is an assistant professor at the Graduate School of Frontier Sciences at the University of Tokyo. Dang. Yes. Shiny. His hobbies and superpowers include, I'm reading what I've got in front of me, general nerdery, sarcasm, and telling biologists why deep learning won't solve their problems. Sorry to most of y'all here. Tonight, Dave is giving us a guided tour of the current genomics landscape. It turns out that what you learned about DNA and genes might not entirely be true after all. Shock. And that understanding the genome is more like playing blindfolded four-dimensional chess rather than stacking Lego bricks. Dave?
Wow, that is very shiny. So first off, a disclaimer. I'm definitely very much under the weather and working through the power of modern pharmaceutics. So if I just collapse, just don't worry. So I'm going to talk about something much lighter than the first talk, namely cancer and math. So the general idea is to give you some very rough pointers on biology. We're going to go very roughly because there's unfortunately a lot to cover. So if you're a biologist, expect to be appalled by the approximations that I'm going to make. And then we're going to talk about why math is actually sort of the same as modern bioinformatics, why they overlap quite a lot. And then we'll try to tie all that together with cancer because it's trendy. So I'm going to talk about how you can cure cancer with math, or at least that's a selling point. Anyway, basically, everything in your body is one of these four things. Obviously, fourth category is fairly vague, but I mean, depending on, for me on a good night, it's pure grain alcohol, but for you it might be different. That being said, it's mostly water, quite a bit of fat, and then proteins, about 25% or so. So protein is essentially how everything in your body works. That's really the important bits. The rest, I mean, water is quite basic. And how is protein made? So protein is essentially how you build everything in your body. Your cells are essentially a bunch of proteins assembled in different ways. And protein themselves are essentially chains of something called amino acids, which are small molecules, and you've probably heard of that. They essentially chain together to make proteins. And guess what? Trivia number one. How many amino acids are there in the human body? Okay. Nope. Trick question. Nope. No? My God, you guys are just. Okay, I can't really hear everyone, so you're gonna have to. Okay, either speak up. So. 21. All right, the gentleman over here is right. Whoever say 21, you're correct. And it's kind of a trick question because there are essentially 20, but and these 20 make 99.999% of all proteins. But then there's that one called cysteine that actually exists in about a dozen proteins, so 21. But as you can see, it's usually 20 common amino acids. Anyway, I had to find a trivia question, so that had to do. So onto essentially how DNA works. Probably all of you have kind of heard of that. The idea that you have DNA and then DNA turns into RNA that's then translated into protein. And the whole thing is about the whole 3 letters codons that make amino acid. So that's sort of the basics of genetics that you probably have heard about in high school or whenever you've studied that. And that's what's called the central dogma of molecular biology. And the word dogma here is one of many, many poor wordings that biologists go with. And there's a bit of foreshadowing here. Namely the fact that, oh yeah, by the way, I went a bit crazy with the transitions because that's my one and only chance. You can't really do that at science conferences. So expect a lot of really cool transitions. You have no idea what Keynote has. Crazy. Anyway, the central dogma of molecular biology is essentially the idea that you go from DNA to RNA to peptides, which are essentially proteins. It's chains of amino acid. And the dogma says it's really like left to right and not much interactions back and so on. The truth actually is that you have a bit more than that. Everything doesn't become RNA that becomes proteins. You have what's called non-coding RNA, which used to be called junk RNA. Once again, turns out it's not exactly junk RNA, so they had to rename it. And the thing about all that is that then it basically interacts in every possible directions. So we are not going to go into the details of that, but suffice it to say, it basically makes understanding the relationship between DNA, RNA, proteins very complicated. Anything can regulate pretty much anything. And that's why it's generally complicated to do genomics these days. So actually, what we call a gene is an interesting question because there's no very easy answer. But generally speaking, it's this. A gene is a region of DNA that is gonna go on. Oh, no. Let's try back. Yes. A region of DNA that's gonna go on becoming eventually a protein. So essentially, only a very small part of your DNA codes for protein. The rest codes for either nothing that we know of or for other bits that then regulate the way DNA is turned into protein. So when people talk about the gene for something, it's generally that bit. And so when people say the gene for eye color or the gene for skin color or whatever type of gene you're talking about, it's not really a different gene. They're all the same gene. They're all, like all individuals, unless they are definitely very badly mutated, share the same gene. What's different is essentially 1 or 2 tiny letters in that gene. And these letters, these small mutations, make the different traits you find between people. There's something called SNP, which biologists pronounce SNPs, which is a lot snappier. So SNPs are essentially how individuals have difference in the phenotypes, their traits. And to give you an example, so let's say you have that one gene, and on one side you have that one letter, on the other side you have that other letter. The way it's going to change is that when it gets translated, eventually it will get translated to a chain of amino acid. And because that letter is different, that amino acid is going to be different too. And then that's going to affect the way the protein folds. And the folding of the protein is essentially its function. Based on the shape it has, it will do different things. So by having that one letter different, you're going to get a different protein, or maybe no protein at all, because it will break down the protein. And therefore, you'll have a different function in the body. For example, and that's a real example with some simplification. Blue eyes are a very simple mutation. They're actually one of the few simple mutations. It's one single letter. So that one is a regular version where essentially you do not have blue eyes. So you have normally functional DNA, and it gets translated by something called ribosomes, and then it turns into RNA that then turns into protein, that then goes on to making your eyes brown or black. So your eyes are brown or black because you have melanin in them. On the other hand, if you have the one letter difference, what happens is that it doesn't really get translated, and instead you don't really get the protein that produces melanin, and then your eyes are blue. It does a few other things, which essentially it helps vitamin D intake, which is why it's a good trait to have if you live way up north, which is the theory of why this gene was selected for Scandinavian people. Anyway, that's for blue eyes. For other things, it's generally a lot more complicated. So all these green rectangles here are genes, and don't even be fooled by how simple that looks. The reality is this is probably very partial, and each of these arrows could mean a lot of different things. So even that graph is a very strong simplification of the actual thing that we probably don't know yet. We haven't quite cracked any pathway, any of these graphs for any serious disease. But the general idea is that something like diabetes, pretty much any very complex disease or part of your function is usually, it does involve dozens, if not hundreds of genes, and they're all interregulating, and some are repressing, some are expressing. It's a complex network, basically. So what we have in terms of DNA and proteins is essentially your DNA is 3 billion letters, more or less in chunks, chromosomes, but at the end of the day, you're looking at this very long string of 3 billion letters that define everything about the way you're going to develop. Then there's environmental factors, but that's, you know, as you've probably heard before, DNA is the blueprint. And about 1.5% of that does become protein. As I said, there's a huge part of DNA that doesn't really turn into protein. It still does things, but we're very unclear on what it does. And that does mean about 20,000 or so genes that become proteins. And then the rest, we really don't know. Quite a few bits we know have a specific function. They regulate, so they will regulate all the genes and decide whether a gene that gets transcribed into a protein or not. Especially in cancer, that actually plays a big role, but we are still very unclear on a lot of that, and that's pretty much where modern genomics is stuck at. So, trivia number 2. So how many in terms of what proportion probably will accept? Do you know how much difference in DNA there are between 2 people? Like any 2 people? Okay, let's do same gender, yes. No, I mean, I'll take either one, really. I'll accept both. Yes, gentleman over here is right. So essentially, you have 0.1% between 2 individuals, but you have, so that's about 3 million letter difference out of 3 billion. However, if you took the whole population, you have actually about 10 million. The difference is that, of course, 2 individuals rarely have every letter different. It's quite unlikely, if not impossible. So what happens is that about 1 in 3 of these possible differences exist between two individuals. So anyway, there's roughly 10 million SNPs, so 10 million one-letter difference, or sometimes a few more than one, but roughly one, out of which about 3 million between two people. But all these letters don't even really code for different proteins, because they're not all in that coding part of DNA. Only about 1% is in the coding part. And on top of that, so we didn't get into these details, but you might remember from high school, there's that code for 3 amino acids, you get 1 amino acid in proteins. And basically, because there's a lot of redundancy, you can switch a letter and still get the same amino acid at the end. So that means only half of these are actually going to result in a difference in protein. So that's roughly that size compared to your entire DNA. So it's a very tiny bit of difference between people, and that's what controls pretty much everything. Why genomics is basically math and not really biology so much. There's about 1,000 reasons and 1,000 subfields of applied mathematics, but we're going to pick 3 in particular. First, noise. Second, the fact that you have partial information. You don't really know everything. And third, epistasis, which we're going to discuss. So, each of these 3. Noise, when you're looking at data, is essentially the fact that you don't have exact values. Your values are usually measured with instruments, and instruments make errors. You can't really see everything. You've got uncertainty, you've got measurement errors, you've got all sorts of technological limitations. So it's not exactly an exact science. You cannot, it's not 1 + 1 = 2. It's 0.85 plus 0.95 equals maybe 2. And so that's a big limitation. It means you cannot really do things with easy algorithms. You need to use generally statistics as a tool to work around noise. The second part is partial information, and that's a huge part. As I showed you before, you have this very complex network of interactions between the different parts of the genome, and at any given moment, you do not stand a chance to measure more than a very tiny fraction of that. So you have to work with this very partial picture of the whole, and you have to make assumptions. You have to figure out how to work around that limitation in order to actually model the whole thing. So to do that, you use what's commonly known as machine learning. Machine learning is essentially the idea of, from a few examples, you're going to try and build the whole pictures. We're going to talk a bit more in detail about machine learning later on. And then the third part is possibly the most important in terms of the difficulty it means. Epistasis. So that's about the only seriously weird word I'm going to introduce. Epistasis is essentially a way of saying combinations. Back in the days, we used to think, or people used to think, that genes were very straightforward. You had one gene, one trait. So blue eyes, gene for blue eye meant that. And that was based essentially on our knowledge of what's called Mendelian traits. So these very specific traits that you can trace between parents. So that's why you know that if one parent has blue eye and the other doesn't, and so on. So these are the easy kind of genetics, the easy genetic traits you can usually trace manually. In reality, though, it never works like that. In reality, you have something that looks a bit more like that. So you have this crazy interaction between group of genes, and that's hard enough as it is, even if you know which genes, but generally you do not actually know which out of your 20,000 genes, which genes are involved in regulating that one trait, which gene means that you will have insulin production or things like that. So just finding this group of genes is going to be a very hard task. And that's basically the power of combinations. If you have 10,000 genes, and we actually have more like 20,000, but let's say you took 10,000 genes. Out of these, if you're looking for just a pair of genes that does something, you already have quite a large number. But then by the time you're looking at 30 genes, if you know that there might be 30 genes involved in the regulation of something, the number of possible combination of 30 genes out of 10,000 is essentially more than the number of particles in the universe. Obviously, you're not going to look at them one by one. So that's where you generally use mathematics quite a lot to, to get a bit faster through your combinations. So here you have it. You have essentially three fairly different subfields of mathematics, applied mathematics and computer science in the mathematics definition of it. They are all very essential to treat genomic data. Okay, so now we're going to talk about what kind of data genomic data is. I'm going to focus on one type of data. There's a lot of different things you can measure, but one of the main things you want to measure is generally RNA. So RNA is that intermediate step between DNA and protein. And it's essentially that messenger thing. It tells you what the cell is doing, what it's producing. So if you find RNA fragments of a certain gene, it tells you that the cell is producing that protein. So it's a way of getting a snapshot of the cell activity at any given time. And what RNA you find in a cell is essentially reflective of what the cell is. Two different cells will have two entirely different landscape of RNA. If you look at a heart cell, a myocyte, and a brain cell, a neuron, you'll find entirely different bits of RNA, because you'll find the RNA that codes for muscle tissue in the heart cell, whereas you'll find a lot of brain-related thing in the neuron one, myelin and other substances. But generally, these are entirely different landscape. And in order to measure that, you essentially you reduce the cells to a fine powder and then get all these little fragments out and then sequence them with big machines are increasingly tiny. And then you get levels. It tells you in your sample that gene's RNA is expressed at this level, whereas this one is at this level, and so on. So it's a sort of a snapshot of the gene activity for the cell. And so from that, basically, for example, you can do these large heat maps that biologists love. And so here, one line is one sample, for example, one patient, and one column is one gene. And the color tells you whether that gene is actually active or inactive. And so right now you can't see much, but let's say we're looking at that for cancer sample. Finally, we're talking about cancer. So these are all cancer samples. So there are tissues taken from either tumor or regular tissues. Blue is cancer, pink is healthy. And on the left, you have basically that snapshot for each sample. And you can even see all the chromosomes up there. So you really have the entire transcribed DNA as a snapshot for each sample. There's probably a few thousand samples. Still not really seeing much. If we try to zoom in by restricting to just a handful of genes, which have been selected because we know they actually have something to do with cancer, so it's been of cheating, but still not seeing much. But if now, there we go. Now, if you actually sort by the type of sample, so here you have the cancer one, here you have the healthy one, you can actually see there's a huge difference between what genes are active in one and what are not in these genes we selected, which conveniently have something to do with cancer. But so that's already kind of a very important idea. It's the idea that by just looking at the RNA makeup, or basically the gene makeup of a sample, you can immediately tell whether it's a cancer cell or not. That being said, being able to tell a cancer cell from a healthy cell is not very hard because it looks pretty unhealthy even just with a microscope. What's more useful, for example, is to be able to tell whether or not your sample will have a recurrence of cancer after treatment or not. Basically, you're trying to tell whether it's a really aggressive form of cancer or whether it's, you know, a hopeful one. So here you got another category where you have the ones that turned out to have recurrence after their first treatment versus one that seemed to be okay. And we are hoping that if we sort that, we'll see differences, but we don't. So typically, the difference between these two is a lot more refined. You have to look at a lot more details than you have to do for just cancer versus non-cancer. But that's a very important question, one of many. But typically, if you can predict that from the sample, then you have a very good indication of the prognosis. So stepping back for a second, why do we care about what genes does what in cancer? Well, first off, because genes are everything. Genes control the way the cell works. They tell you essentially how the machinery sort of interact. So by looking, if you know what genes are doing something, then you can figure out what is the cause of the cancer, what is the gene at the root of everything. That's the understanding part. And the treatment part is that if you actually identify genes that are uniquely tied to your cancer cell, then you can try and target them. That's not even quite where we are at in terms of current treatment. Right now we are targeting very large compounds, but the general direction is that eventually we find exactly what genes are involved in the cancer and then we can target that. And so how do we do that? How do we try to figure out from this large heat map of gene expression which ones are, let's say, recurrence and which one are full remission? So you could plot these levels. So that's like one gene that we plot on one line. And if you show which one are recurrence, one remission, then you can see sort of an easy frontier, and then you could tell, well, okay, if gene A is that level, you've got a good chance of remission versus recurrence. Now, if you're looking at two genes, you get something like that, and you can try and do a separation. The problem is usually you have a lot more samples, and then your separation no longer looks very good. But then you can adapt it, but then you get more sample, and it still doesn't look good at all. And that's where machine learning comes in. And one important lesson of machine learning and something called transduction is that you should never try to solve a general problem when you're trying to solve a specific problem. And I find that a very good lesson in life too. So the idea here is, instead of trying to really do perfect classification, it turns out that if you just do that, you actually get a fairly good classifier. It actually works very well, at least the best you can. So that's what machine learning does in general. It tries to infer what the real data might look like from a few examples. Now, if we have 3 genes, we could try plotting it in 3D, and then good luck trying to find that. But then if you have hundreds of genes, try and imagine something in hundreds of dimensions. That's where usually math comes in. And that's how you reduce that problem to something that can be figured out. And that's how you plot that, and then math tells you, actually, these are the groups. And so the green one are healthy cells, the red one are known unhealthy cells, and the rest is kind of unknown. And by doing that reduction, by plotting it, we know that the cancer is around there. Okay, very quickly, what causes cancer? Because that's what you're all here for. Obviously, we all know cats cause cancer. Still waiting to hear from the Academy of Science on that one. The second theory is that it's mostly bad luck. Cancer is essentially random mutations that win a lottery ticket. And allow me to illustrate very quickly. So that's a vague idea of what your cell might look like. It's actually way more than that, but generally speaking, like that. And you have bits of it that do important things, such as repairing your DNA. Your DNA is actually self-repairing. Otherwise, it would very, very quickly degrade because DNA is very fragile. So you've got this bit that repairs it and that bit called apoptosis. And that's the last trivia question. What is apoptosis? Okay. Yeah, I'm not sure who said it first. You'll have to figure it out. Okay. Okay. So these two parts are very important because so in everyday life, your genes or your DNA gets attacked all the time. I mean, if you go in the sun, your skin cells get, destroyed all over. And that's fine, because once the DNA repair mechanism no longer follows, eventually the cell just is no longer fit and it will die. And that's fine. The problem is, every once in a while, the bits that get attacked are the very bits that are supposed to fix the cell, the DNA repair mechanism, and the bits that are supposed to make the cell die. So if these two bits get attacked in either order, but eventually get attacked, and it takes a lot because they're very recurrent mechanism. It's not just one gene, it's many genes that have to be dead. If these bits get attacked, the cell no longer dies its natural death, and you get a zombie cell. That's not yet too bad. That's essentially a benign tumor. The problem is, at that point, you might also attack the part that regulates how the cell replicates, mitosis. And if you also get that destroyed, which will happen a lot faster once these two first bits got attacked, because once the DNA is not self-repairing anymore, all the mutation accumulate a lot faster. And then once you actually also have a cell that has both this lack of programmed cell death, this lack of self-repair, and starts replicating very fast, then you do have a problem. You have a cell that starts taking over. And that's essentially what cancer is. Essentially, it's a few mutations that hit the right spot and then start really peaking up. And once you actually reach that point, once the cell is replicating and mutating, that's when you have a malignant tumor. And on top of that, at some point, while still hitting all these random points in your DNA, you're also going to disable differentiation. So that's part of your DNA. The DNA is what makes one cell belong to one tissue, one heart cell versus one brain cell. And by triggering the right genes on purpose or through cancer, through random mutation, you eventually get what's the equivalent of a stem cell. That's why a lot of people usually talk about cancer being like stem cells, which is a bit dodgy, but the general idea is that at some point, once your cancer cell is no longer differentiated, then it can just start circulating in your entire body, and that's what's called metastasis. So essentially, up to that point, your cancer is limited because it still has to belong to the tissue it belongs to. After that, it can go anywhere, and that's where it gets a lot more difficult to treat. So the future, definitely not quite the present yet, but the future is essentially to find these original cells, because the problem is right now what we do is we look at cells that are generally cells that are generally different, and we try to remove them either surgically or chemically. And we hope that we are removing all the bad cells. But generally, you leave these early mutated cells, which are also the worst one, because they're the ones that have the power to mutate again into another cancer cell. And that's why cancer is very often recurrent. So ideally, you identify the very early cancer cells, the first ones that actually triggered enough mutations to become cancer cells. And then you target them, as I said, by specifically targeting one gene or another gene that is specific to that cancer cell. And so what you want is to identify the lineage of these cells. So this data set, in this one, each line and each dot is one cell. So that's a fairly recent technique. Until now, we couldn't really measure one cell. We had usually a bunch of cells together, averaged and measured as a whole. Nowadays, we can actually measure one cell at a time in a whole tissue. So what we can do now is take a bunch of cells from your tumor and then measure them, and then try to find what the order of the cell is. So obviously you don't have all the cells, you don't have the whole family tree, but using math, you can generally find the relationship that exists between the cells and try to figure out which one came before which one. Generally because the earliest one have less mutations, and also because the RNA landscape looks a bit more normal, less like a crazy cancer cell. So the idea is from that, you want to try to do that kind of stuff. So that's actually what I do. One of the things I do. It's making cell tree out of cell data. So that's for neuroblastoma, which is actually not a real cancer, more like an autoimmune disease. And the interesting thing about neuroblastoma is that, so neuroblastoma is essentially brain cancer. And also mainly kids. So generally kids between 6 months and about 6 or 7 years tend to get neuroblastoma. And the crazy thing about it is that it has a very poor survival rate unless you're one of the lucky people that gets full remission regardless of treatment. So essentially we don't have any good treatment for neuroblastoma, but in about 20-30% of cases, there's just a normal, the body fights it on its own and you get full remission. So obviously there's a difference between the patients that get full remission and the one that don't. And it's not a freak accident, it's something like about 20-30%. So it's very important to understand what's the difference is. And that's what happens with these cells. We have a bunch of different samples. We know that some of them will survive and some won't. We don't know which ones because we have the samples early on. Because if the tumor had disappeared, then we no longer have anything to measure. But let's say we took 10 patients, we are nearly guaranteed to have 1 or 2 patients that will eventually go into remission. And just a little thing, it's actually not humans. We're talking about mice here, but same thing. So what we know is that some of these cells are good cells that will survive neuroblastoma, some are not. And so by having that tree and, looking at a few more details than what we know of the way cells work, we know that one branch is a bad branch, the other branch is a good branch. And then, we have to figure out through other methods what happens at that crossing. And, basically that's the whole idea of the treatment. If we can figure out the difference, try to, target the genes that are specific to the bad branch, or make the bad branch more like the good branch by triggering the right pathways. That's my last slide. And the takeaway message is that essentially everything in your body is genetics. It's not just, your height or things like that. The way cells work is through genes. And these interactions are very complicated. Cancer, in particular, is an aspect of the way these genes work, and math is essentially how we understand all that. Thank you, that's it.